Good morning.
Yeah, and I heartily welcome you for this webinar. And let me give you a little introduction about the group, the audience which will be listening to you. You know, we have a bovine embryo transfer group, which consists about 90 members. I'll share with you the directory of that soon. And they are the mainly working into the some or other aspects of in vivo embryo production or IVF. Then we have a very strong society known as Indian Society of Animal Reproduction in the country, wherein majority of the veterinarians who have done their masters or doctorate in gynecology, reproduction, they are associated with that, and they are connected in some or other way with the embryo work. So you have, everybody is very keen to hear you, and I would like to formally welcome you for this webinar, and really grateful to you for accepting our invitation on behalf of Bovine Embryo Transfer. And I represent the Bovine Embryo Transfer Group also, and I'm also as a national mentor with the Ministry of Animal Husbandry, Government of India. So I also welcome you on behalf of the Ministry of Animal Husbandry, Government of India. And we, in our committee, we have a central monitoring unit for regulating the activities of the embryo transfer in IVF in the country, and that has been constituted by Government of India, and I'm as the chairman of that committee, and we have another 5 members of that committee. I'll introduce to you later. To everyone, I would like to give a brief introduction of Dr. Jennifer Barfield. She completed her bachelor's degree in animal science from North Carolina State University in 2000, and then she completed a doctorate in conservation biology at the University of New Orleans, where she studied potassium channels on sperm as potential target for a non-hormonal male contraceptive. This work was completed in partnership with the University of Münster, Germany. Following her doctorate, Jennifer completed postdoc in Colorado State University and spent 6 months at the USDA's National Animal Germplasm Program, followed by 2 months at the New Bolton Center at the University of Pennsylvania before returning to Colorado State University as a faculty member in 2011. You know, she is currently an assistant professor in the Department of Biomedical Sciences, where her research focuses on assisted reproduction, primarily in bison and cattle. She also co-founded a professional master's program in assisted reproductive technologies, which began in August 2013. Her bison research significantly contributed to the establishment of the Laramie Foothills Bison Conservation Herd in northern Colorado, which she continues to manage. Jennifer, I would like to definitely tell you about my memories visiting the Colorado State University, but well back in 1985, when I had spent some time. That was almost, I think, 37 years back. I spent some time with the Professor George Scheidel. I still really remember him every now and then, and Sarah Scheidel also. So giving you a little background about our Indian scenario, India is the largest milk producer in the world, but the per production capacity of an animal is very low. It's only to the tune of about 1900 kgs per lactation. And as you know, in India, cow is a sacred animal. So we have a tremendous potential of using nowadays with the sex sorted semen, because we are more interested in getting the female, because we don't eat the beef in India, a majority of the country. So with that, and then there is a tremendous potential for improving the allied animals. And as you know, India is an excellent source of Bos indicus breeds, and from our country, the animals like deer have gone to Brazil, and now the time has come for us to import the semen and embryos from Brazil. So we need to do it. We can do a lot, particularly into our indigenous animals, and definitely we have imported the Holsteins and Jersey. So various breed work has been going on across the country, and there is so much of potential. No doubt, IVF is only in a very initial stages. Our team members have done a lot of work on the MOET, and gradually knowing the importance of the IVF now, we are shifting it, and Government of India has funded almost about 20 labs in the country. A good amount of funds have been given to the universities and livestock development boards. And there are about 4 to 5 private organizations also, which are working into the IVF. The only thing is that it's really still an expensive, because as you know, I mean, I did not tell you, but the main problems occurring is that we need to get a good number of embryos, good number of oocytes converted into a good number of embryos per OPU, and then, need to get a good pregnancy rates. And we do have different problems with the recipients. It's not like organized recipient farms are there in the country. So we do have a different tough conditions in selection of the recipients also, because we don't have a large commercial farms wherein you can get a very good results like in Brazil and United States. And we have been very lucky to have a lot of speakers from Brazil, from United States for the last few months, for the last 6 months. And again, we are really grateful to you for joining this webinar and addressing us. I will quickly, let me introduce you just in a few seconds. I mean, to my members who are there. Let me start with Dr. Sanjeev Chaubal. Is he joined from Sanjeev? Is Sanjeev, Dr. Sanjeev Chaubal?
Yes, he joined, I think.
He has not joined. Okay, I think there is some problem here. Jennifer, Sanjeev Chaubal is in USA, and he's basically from India, but he's working for Transova. He's a manager with the Maryland lab, and he's one of the core committee member. Then we have a second member, Dr. Mutharao. Dr. Mutharao, just say hello.
Hello, ma'am.
Yeah,
good morning.
Dr. Mutharao also has been doing a lot of work for almost more than 2 decades now in MOET and IVF now. Then we have a Dr. Siddharth Laik. He is with the National Dairy Development Board. Siddharth?
Hello, hi.
Dr. Siddharth?
Hello.
Yeah, he is Dr. Siddharth.
Yes, hello, sir.
And he is with a very important organization known as the National Dairy Development Board. So he is a manager of the IVF lab over there. And then we have Dr. Ajay Aswal. Dr. Aswal, can you just say quickly hello?
Hello.
Yeah, Aswal, I think you are driving right now. Yeah, okay. So Dr. Aswal is there. Again, he is almost more than 2 decades into this work. And we have fifth member, Dr. Amol. He's traveling right now. So is Sanjeev joined? I mean, Siddharth, you help him in joining and send a
yes, he joined.
I joined.
Okay, Sanjeev, say hello to you. No problem.
Yeah,
you have heard her.
Yeah, I have heard her. I have met her also a couple of times. Hi, Jennifer.
Hi.
Yeah, I have also met Jennifer. I have also met her. So we know, at least 2 of us know her. Good. And with that note, Jennifer, I request you to kindly start your talk, please. Thank you.
Well, thank you. First of all, I'm really honored to be able to speak to this particular group today. And thank you for giving a lot of that background on the groups that are joining us today. And yeah, it just gives me a very good appreciation for what you are doing and the challenges you may have and kind of where you are in the process of producing IVF embryos. So hopefully what I will present to you today will be helpful. And I'm happy to answer questions. There'll be a portion of this where if people want to reply through chat, I will try to keep up with that. And we'll go through some pictures and things later. But just really honored and grateful to be able to talk to you all today. So thank you for that. And yeah, so it's morning here in Colorado, and I just want to say, first of all, that I have about an hour presentation, and then we'll just see how the discussion goes. But yeah, we can kind of delve in from here. So what I thought I would go through today are really just reviewing some of the basics of embryo grading, why we do it, how we do it, what are the criteria, and all of this will be based on the IETS standards. So And then once we kind of go through those basics and I show you some pictures, I have a group of photos and videos that we can look at together to look at some of the different morphologies and how that might impact how you grade an embryo. And then I'll just speak a little bit about the future and where we might be going. So first of all, why do we grade embryos? I think it's really important to acknowledge right up front that grading embryos is a subjective process. And in that, it means that it is imperfect. Not everyone will always agree on the grade of an embryo, but it is our easiest method currently to be able to evaluate an embryo's potential for creating a pregnancy and ultimately a healthy offspring. In addition, grading embryo really helps us to monitor how our IVP systems are working. So if we have kind of our standard embryo quality that we see coming out of our IVF system, if that drops or changes, it can indicate problems very quickly. You might still be making embryos, but they might not be as good a quality. So, that's something that you want to keep monitoring. And also on the other side of that, if you start to get really good quality embryos, they are looking better than normal, then you can go back and try to learn something from what has changed. And so having that benchmark for the quality of embryos that you produce through your system is really important. And then for those who may be doing research, of course, it helps create continuity in our publications so that we're all grading embryos in a fairly similar way so that data can be evaluated on a common basis and we can really start to tease out things that are working and things that are not in the research that's going on for in vitro production of embryos. So of course, it's probably not a surprise to this group that, grading the grade of an embryo really does correlate with the pregnancy rates that you may see. So these data are from in vivo produced embryos, and you can see here we have the IETS grades along the bottom here, and the pregnancy rate from an an actual 9,021 embryos. These are older data, but the trends with getting high pregnancy rates with grade 1 embryos and that stepping down with every decreasing grade remains true today. And there's no evidence that in vitro produced embryos behave any differently. Now, I will say that they do have pregnancy rates with a grade 4, and this was off of a 5 scale. So it's not exactly true to IETS, but because typically for IETS 4s now are degenerate or non-viable. So, but you would still see this step down process. And we'll go into a little bit more of those grading systems in a bit. So we do have some data from in vitro produced embryos, and this is actually from dairy cattle. And here what you can see, we've got grade 1 embryos and grade 2 embryos. And these are pregnancy rates from embryo recipients that were either heifers or lactating cows. And here you can see really just the quick trend is that grade 1 pregnancy rates were about 50%, and grade 2, they were right around 30-35%. So again, grading is important in that it can give you some estimation or, some idea of which embryos would be better for transfer and likely result in pregnancies. Now, grading embryos is not a new thing. This is a paper from 1995 that looked at a variety of different features of embryos that may impact the grade. So some of those would include the shape of the embryo, particularly the zona pellucida, the color of the cytoplasm, which can indicate the density or the amount of lipid. Now that might vary a little bit from species to species or breed to breed. And so it's good to know what you're working with there. The number and compactness of the cells in the embryo. And so that might be important for different parts of the embryo. So a morula, kind of the whole compactness. And then for a blastocyst or an expanded blast, the compactness of the inner cell mass. The amount of PV space, perivitelline space, if you are evaluating a stage of embryo that should have PV space. The number of extruded and degenerate cells. frequency and size of cytoplasmic vesicles, and then of course the stage of development relative to age. And we're going to go through all of these with some pictures and in a little bit more detail. Okay, so just as a reminder, the IETS scale is a 1 through 4 scale. A lot of times in the literature, particularly some of the older literature where they may have not adopted the IETS grade standard yet, this is generally how those different grades correlate. So for IETS, they've really collapsed the excellent and good quality embryos together into one category. And largely that is for import-export reasons. Good quality embryos still do produce higher pregnancy rates. And so that was just to make sure that people weren't refusing the import of embryos that had really good potential for creating pregnancies. And again, as I mentioned on the previous slide, degenerate embryos in the IETS system are 4. In some of the other systems, it's a 5. So the IETS grading system has a variety of components, not just, the specific morphological features of the embryo, but also how much of the cellular material within the zona pellucida is part of the viable cell mass, okay? So, or what we often call the embryo proper. And so for grade 1, there's a cutoff where at least 85% of the material within the zona must be part of the embryo proper. So a grade 1 embryo does not have to be a perfect embryo. So there are some examples of grade 1 embryos here. Particularly in this first one, you can see this very large extruded blastomere from the early cleavage stages of the embryo, but otherwise this embryo looks fairly healthy. So it would still be considered a grade 1. Now that benchmark kind of lowers significantly when you go to grade 2, where only half of the material within the zona pellucida needs to be part of the embryo proper. And you can see likewise, it drops down to 25% for a grade 3. And then of course, a 4 is a degenerate. And so there would be no part of that embryo that would be viable. Now, part of grading embryos accurately is staging the embryos correctly, okay? So this is from the IETS manual, this drawing here on the right, and you can see all of the different stages that they recognize and how they divide those out. And so many times, at least here for bovine practitioners in the US, these numbers that are before the embryo stage, those are often used as codes on paperwork. So if you have a morula, you would call it a 4. If it's a grade 1 morula, it would be a 4-1 on the paperwork. So I just wanted to make sure that you had that reference if you wanted it. And for particularly if you're looking at research and they're using the numbers instead of the names of the stages. So that's there for your reference. But a morula, of course, should have some perivitellin space. So that's this area here. And it should be nice and compact. You should be able to resolve a line essentially of those cells that are all connected together and kind of compacted down. And it's very difficult sometimes to pick out morula, particularly when they are of poor quality. That's one of the hardest things that my students do. One of the things they struggle with the most. But this is really just a beautiful, perfect morula. There may be a couple of extruded cells here, but just a gorgeous, gorgeous morula there. Now the early blast also has perivitellin space, and typically, according to IETS standards, the blastocele cavity is 50% of the embryo or less. Okay, so this one is about 50-50. Probably the ICM is still a little bit larger than the blastocele cavity here, which is why we would call that an early blastocyst. Now, when that blastocele cavity gets larger than 50% of the embryo proper, that's when you begin to call it a blastocyst according to IETS standards. And in our lab, when we're grading students, and that can be a very subjective thing, we tend to just call a blastocyst an embryo where the blastocele cavity has expanded all the way out to the zona pellucida, and there is no more perivitellin space. And then for the expanded blasts, the real identifying mark here is that the zona pellucida is thinned. So if you look at these earlier stage embryos, you can resolve a very thick zona pellucida, but for the expanded blast, it's very, very thin. And the outside looks smooth, okay? And so that's really important to be able to resolve when you are trying to discern an expanded blastocyst from a hatching or a hatched. So here you can see the zona pellucida is broken here and the embryo is coming out, and you can see this bumpy nature. You can see actually individual cells of the trophectoderm. And then for the fully expanded blast, it looks completely bumpy all over. So it's really important that you are able to see the distinction between kind of that smooth surface of the expanded blast to be able to classify a hatching or hatched blast appropriately. Now for IVF embryos, it's very important when you are considering the stage, because that does have an impact on what grade you give the embryo, that you're not comparing it directly to an in vivo system. And that's because what we call day 0 in an in vivo system is different than what we call a day 0 in an in vitro system. So this particular figure shows the progression of there's the oocyte, it's immature, it's mature, and then embryo development, where it would occur in the female. And then this is kind of the matching in vitro system. In our lab, we use a 2-step culture system. But here you can see day 0 is often what you call the standing heat, okay? And so ovulation is gonna occur later, and meaning that fertilization will occur significantly after what you're calling day 0. Whereas in the in vitro system, you can see day 0 is considered the point in time in which we put the sperm and the eggs together in a petri dish or a 4-well dish and begin that process. So fertilization is going to happen rather quickly from that point relative to the day 0 of an in vivo system. So what that means is that when you are evaluating a day 7 embryo, the stage of development that you expect is going to be different for an in vivo produced embryo versus an in vitro. So in an in vivo system, you are mostly going to be seeing morula and early blasts, though you can see blastocysts, because especially if you are superovulating, you might have eggs that are being ovulated at different times, and so development is going to be a little bit different. But on your in vitro system, day 7, you're mostly going to see blastocysts and expanded blastocysts and potentially even some hatching. Let's see. All right, so I will try to get to some of these questions in just a little bit. I'll get through the stage portion first, and then we can talk about the day 8s. So thinking about stage of development and how that affects the quality score that we give them, the grade that we give them. When we look at the embryos, they are at the expected stage of development and they're a perfect embryo, then they get a grade 1, okay? Now, if it's a perfect embryo, but it is delayed by 1/2 to 1 day, then you would downgrade it to a 2. If it's behind in stage from 1 to 2 days, so typically in an IVF system, this might be like a pre-compaction morula, then you would give it a grade 3, even if it's a very pretty embryo, there's no extruded cells. And then if it's more than 2 days behind, you would consider it degenerate. So just to kind of put this in a table form for comparison, if we're looking at in vivo derived embryos on day 7 versus in vitro produced embryos on day 7, a pre-compaction morula, assuming everything else is perfect about the embryos, this would be grade 2 for both systems. Now the difference comes here in the morula early blast stage. So morula in an in vivo system on day 7, perfect embryo grade 1. Perfect morula on day 2, in an in vitro system on day 7, you would give it a 2 because it's behind in development. Now I put 1 or 2 here for early blast. It really depends on what time of day on day 7 and how often you see early blasts. In our lab, we tend to give early blasts a grade 1 on day 7 if they look perfect. But then in both systems, you get 1s on blasts and expanded blasts on day 7. So stage matters. So let's talk about what other features may impact an embryo's grade. Okay. So one of the things I mentioned a little bit earlier was compaction and perivitellin space. So I will say that as our IVF systems have gotten better, we have seen fewer problems with compaction in IVF systems, though it still exists. And so these two pictures are examples of loose compaction on one side of a morula. So you can kind of see this very distinct line, and then right around here it fades out and you see there may be some cells that are being extruded here. In this one you can see this very bumpy edge and a little bit of looseness here compared to these in vivo derived embryos where you can see this very clear line all the way around the embryo proper. So in this one it's just really fuzzy and you can see there's an extruded cell here. And so this would be an indication of a poorer quality embryo or poorer quality morula in this case in an IVF system. So that can be one problem you might encounter, or one morphological feature that might be a reason to downgrade, particularly a morula. Now vacuoles are really interesting because in IVF systems, and sometimes in vivo derived systems too, you see embryos with these small little vacuoles. Sometimes they can be rather large. Of course, this is kind of the extreme end of that scale. And for many, many years, we have talked about vacuoles as a reason to downgrade, particularly when you get these very large vacuoles in the embryo. But, you know, we actually don't know because we don't often follow these embryos and whether or not they create pregnancies. So I had reached out or had a conversation with Daniela Demetrio. She is a bovine practitioner here in the US who works on a dairy. And she says that in her experience, there are some cows that just produce oocytes with vacuoles. And those oocytes do make embryos that also have vacuoles. And while she hasn't published this or studied it, you know, kind of set up an experiment and actually gone through the study, she does have recorded pregnancy rates of up to 60% when she has transferred embryos with vacuoles from these cows that have oocytes with vacuoles. So I think the jury is still out about whether or not these vacuoles really do impact embryo quality. We don't know what causes them. And so we don't really know how to prevent them. But if you're getting 60% pregnancy rates, despite the fact that there are, as you can see here, potentially some larger vacuoles in the embryo, it really makes you think, is this really a reason to downgrade an embryo? So yeah, we don't know what causes them. Another interesting observation is the shape of the inner cell mass. So in IVF systems, we do see a lot of different morphologies. And so here on this right side, you can see that we have these compact inner cell masses. Okay, so they're very easy to resolve, versus on the left side here, the inner cell mass is stretched out across the embryo. Now, it's really important that you roll the embryos. We'll talk about that a little bit more later to, you know, make sure that this embryo isn't sitting in just a way that the ICM is stretched out, but you've got it rolled so that it looks compact. So you need to make sure you look at it from all angles. But the question is the picture on the left, is this abnormal? Is it an artifact of the IVF process? Is there something not going on correctly here? And we don't really know the answer to that either at this point, but we do see these different ICM morphologies. And I thought this was a really interesting picture. This picture here was sent to me by Dennis Avery. He's a veterinarian here in the United States, and he flushed a cow and he was expecting on day 7 morula and early blasts, but he got this beautiful selection of expanded blasts and blasts. And what I noticed about all of these in vivo derived embryos is that their ICMs are kind of more similar to this particular morphology of the ICM in this in vitro produced embryo here, which, if we are trying to create a system that most closely mimics the in vivo environment, then perhaps this is really the morphology of the ICM that we want to be looking for and considering those as our top quality IVF embryos. So again, it's been really hard to kind of track these embryos, look at all these different features of the morphology, and then follow it out to pregnancy. But I just thought this was a really interesting observation. And you don't often see a group of in vivo derived embryos at this stage, at least not such a big group. One of the other things that has often been a question when I've attended conferences, the American Embryo Transfer Association and the Canadian Embryo Transfer Association, and even with IETS to some extent when we've talked about embryo grading, is should we downgrade for a collapsed embryo? So in this, on this slide, what you can see here is an expanded embryo. The zona is thinned out, and the embryo proper has collapsed away from the zona pellucida. Now, because we have all of these time-lapse incubators now, we can watch embryos during the course of development, and we see that as the embryos are expanding, they do have this pulsing action where they will grow, they will push out against the zona, and then they collapse down, and then they come out again, and then they collapse down. And so the feeling that I get from most of the practitioners that I've talked to, and I have actually polled them during sessions when I'm giving live seminars, is that most people do not downgrade for collapsed embryos. So we think it's part of just that natural expansion process. But I will say that a collapsed embryo does give you the opportunity to see any extruded cells that might not be easily resolved when the embryo is expanded all the way out. And pushing up against that zona. So it really does give you an opportunity to understand if the embryo is of poorer quality. And here on the right, you can see some hatched embryos that are either expanded or collapsed down. Now, it's very, very hard to grade a collapsed embryo that's totally hatched. You can see, at least just in these examples here, some of them appear to be more intact than others. This one seems to be a little bit more diffuse, but you can see that there are cells coming off and being extruded from this embryo actively. So these are very, very difficult to grade. And these are just some other pictures of some hatching, these three here versus hatched embryos. And so these are not necessarily fully expanded. So this one is still partially collapsed, but it looks like it's a very good quality. You can still resolve the ICM versus some of these others where the ICM isn't necessarily as clear. And in many of these, you have some extruded cells, but nothing significant. These are in general pretty nice hatched blasts. All right, so one of the other things that we do look for, as I mentioned, is the amount of cells that have been extruded from the embryo. So these two pictures are to demonstrate what it looks like when an expanded blastocyst has a significant amount of extruded material. And it can be very, very difficult to pick out, especially if you're looking at an embryo at low magnification. So in this embryo on the right, we have very few extruded cells. So you can see the inner cell mass is easy to resolve. You can see the trophectoderm, the cells of the trophectoderm are touching the zona pellucida. They appear to be very uniform in size with no major disruptions or abnormalities. If you look at this embryo on the left, the inner cell mass is actually sitting on the bottom of the embryo here. So this is your ICM. But from a distance at a lower magnification, you might think this is one of those spread out ICMs. But really what this is are cells that have been extruded or kicked out of the embryo essentially and now squished against that zona as the embryo is expanding. So this would be an indication of a lower quality embryo, and still the IETS guidelines for the percentage of that extruded material would go into the grading of the embryo. These are just some different stage embryos where you can see some extrusions. Here on the left, this is a morula. It's not as nice and compact as we would like, but you can see there is some extruded material here in the perivitellin space. And likewise on this early blast on the right, so here I'm outlining the embryo proper. You can see the blastocele cavity in here, but all of this material out here is extruded and not part of the embryo proper. So again, it takes a pretty keen eye to be able to pick these up, and really requires you to move your embryos around to get a good idea of what's extruded and what is not. Okay, so if we put all of these things together, let's go by stage and grade and look at some different examples here of first, the morula. Okay, so grade 1, these top 4 embryos, they're not all perfect, but for the most part, you can see that they are nice and compact. There's a little bit of loose cellular material on the second one, but there's very little extruded debris. The 4th one probably hits that limit of about 15% being extruded. And the full morula is a little bit obscured by the debris that's sitting here on the top. So grade 2, you can see there are there's significantly more material outside of the embryo proper. So in this first one, the top part here is your embryo proper. This is an extruded blastomere. Here is the embryo proper for this one. These are extruded blastomeres. This one, the morula is here, and you've got 2 areas of extruded material here. And then in this one, you've got your embryo proper here, and then a bit of material that's not in the embryo right here. Now, grade 3 is when it gets really tricky. This is what my students really struggle with, because this really involves moving the embryo around to be able to see it from all sides. So for this first embryo, the embryo proper is right here, and all of this other material is junk, okay? In this embryo, and this one's probably the hardest to see of these pictures, the embryo proper is in here, and it's obscured by material that's not part of the embryo on top and here on the side. In this one, again, really hard to resolve, but the morula was right here, and this is all debris here. And this one I actually took from the bovine ova tutorial for IETS, because here you have a very small morula. This is the cellular mass here. You have a large blastomere here, and then you have a bunch of other material that's not part of the embryo proper sitting on the bottom of the embryo there. So this gets really tricky for people to see, and if you don't have a good microscope, it can be very difficult to resolve the difference between these 2 masses, recognizing that these are your live cells. Okay, for the blastocysts, so I will say the zonas are a little bit thin on some of these. This one's a little thinner than I would like to call a blastocyst, but you can see there's a longer inner cell mass. Okay, so we haven't decided whether or not that's a good thing or a bad thing, but you have a little bit of debris out here for this one. You've got a clear inner cell mass and blastocele cavity for this one, and so you know, the ICM can be, you know, still close to 50% of the material, but that will become a less, from a percentage-wise standpoint, less of the embryo as it gets larger, okay? For grade 2, again, more extruded material, some darkness. This is just extruded material that's pressed up against the zona. This one has some irregularities in the blastocele cavity, it looks like, and not a very well resolved ICM. And this one has some vacuoles, and it just looks, you can see right in here, some of these spots where you have some vacuolization. Now the grade 3s, in these embryos in particular, it's very hard to resolve clearly the ICM. So you kind of think you know where it is, but a lot of times it can be obscured by a significant amount of extruded material. You can get these kind of dark cells, speckled cells throughout the embryo itself. And so you can see there's just really significant abnormalities in these embryos where you don't have a clear ICM like you see up here in the grade 1 row. Same really for expanded blasts. We're just looking at, you know, kind of a larger blastocele cavity, thinner zona pellucida. But what you really want to see in these embryos, particularly for them to be a grade 1, is this really nice compact inner cell mass. In the grade 2s, it's a little less organized, so you can have abnormalities in the trophectoderm. So here you can see there are some irregular cells and vacuoles in the trophectoderm of this embryo, though it still has a decent inner cell mass. This one you can see there's a large amount of extruded material, That's been pressed up against the zona. Here is your inner cell mass. And the same here, it's just not as nice and compact as these that you see on the top row. And then when you get to grade 3, again, kind of just like the blasts, it's really hard to resolve. The ICM, you've got major abnormalities in the trophectoderm. So kind of both the ICM and the trophectoderm are flawed in some way. And then you can either have some really significant amounts of extrusion. So I just wanted to show these pictures while we're talking about blasts and expanded blasts of things that are not viable. And these can be really tricky to the untrained eye because these are just, large vacuoles and fluid-filled spaces that are not blastocele cavities. And it can be very tricky to pick these out from, an embryo with a blastocele cavity, particularly a poorer quality embryo where some of the extruded material may be obscuring the embryo proper. But the key to discerning these types of embryos from, say, a grade 3 blastocyst is looking at kind of this smooth line of these fluid-filled spaces. Okay, so obviously these 4 vacuoles are really abnormal. But like if you look at this first embryo and you see how smooth this line is, that has a very different appearance to an actual blastocele cavity that has trophectoderm cells around it, which will appear a little bit more bumpy. So let's just go back. Here, you can kind of see the bumpiness of the trophectoderm here and along the edge here. I know this picture is a little bit small for you to see that on, versus this very clear, smooth line in these abnormal, non-viable embryos. So for hatching hatched, grade 1, I actually didn't have a lot of pictures of grade 3 hatching or hatched. So I don't have those to show you, but you can see clear ICM, healthy trophectoderm cells. They're all uniform in size, more or less. Very few abnormalities or dark cells. This one is leaving a blastomere that stopped dividing behind. That's totally fine. The rest of this embryo looks really beautiful. And then the grade 2's poor quality ICMs, some abnormalities in the blastocele cavity for trophectoderm. And this one's just a little dark. It looks like it's a little bit unorganized. I'm not so worried about the amount of extrusions here. That's pretty minimal, but it just looks a little dark and unhealthy. So a lot of these things can be a little bit tricky to pick out, but I will say the key to doing it is to do it a lot, and to be able to do it well is to do it a lot. And so I always like when I talk about embryo grading, because not everyone's going to agree. And even within your lab or your group, you will disagree. And that's completely normal in a subjective process like this. There was a study done in the 90s where they took very experienced embryologists, I mean, decades of experience, and they had them not only stage embryos, but grade them according to the IETS scale. And they compared in vivo and in vitro produced embryos. Now, I think they did this off of pictures only, so just to caveat it there. But for stage, you can see that this is the percent of agreement by the embryo evaluators. So for the stage, they agreed more or less for morula for both in vivo and in vitro. For the blastocyst, there was slightly less agreement for what was a blastocyst for the in vitro. And everybody agreed on what a dead in vivo derived embryo was, but there was some variation for the degenerate in vitro embryos. But when it came to grading, you can see that these values drop significantly, and really where it's difficult is here in the 2 and 3s. Okay, so those can be really difficult to resolve at times because you've got an embryo that doesn't look great, but maybe there's some good features, maybe there's some really bad features. And so this is where you get the most disagreement, even among very, very experienced embryologists. Okay, so practice makes better, but not perfect. That's what I'm going with here. Okay, so this was a poll that I took during an American and Canadian Embryo Transfer Association meeting. We were doing all these little eye clickers where I would show pictures and things. And I thought this was a really great example of how years of experience can really influence your ability to grade embryos consistently. So I showed this picture. I said it was a day 7 in vivo produced embryo. And what grade would you give it? Now, day 7 in vivo, we would expect morula early blast. This is a beautiful early blast. It is a grade 1 all day long. And you can see here that the group with the most experience, so 20 plus years of experience, 92% said this is a beautiful embryo. And that percentage declined down to the 60s when we got to 10 years of experience or below. And some people said this was a degenerate embryo. Okay, so only really 2 of the 19 people in this less than 5 years of experience group, but still there's nothing wrong with this embryo. So looking at a lot of embryos and grading them regularly is really the key to becoming better at this and being able to select embryos that are going to give you higher pregnancy rates. A couple of other tips that I'll mention, and I think I've said this a couple of times already, is that you have to roll the embryos. It's very hard. We're going to look at pictures here in this seminar, and you're going to look at pictures in other seminars, but to really grade an embryo appropriately, you have to roll it around and see it from every angle. Sometimes a blastocele cavity is hidden under the ICM and you have to roll it over to get the stage right. Other times you have to roll embryos to make sure that you can see all the extruded material, so you're grading it correctly in that way. And so that's very, very important. The magnification also matters. We recommend at least a 60x magnification on your stereoscope to be able to resolve some pretty important differences. So in this picture here on the left, this of course are 2 embryos at a fairly low magnification. And these are the same 2 embryos at a higher magnification. And what you can resolve here that you cannot really resolve here is that the one on the right is a grade 1 morula and the one on the left is an unfertilized oocyte. So if you were just looking at them at a low magnification and you said these are 2 great morula and you transferred these 2, you'd be wasting a recipient on the one on the left. So it's really important that you not only roll the embryos, but that you increase your magnification to be sure you're grading it correctly. And then the third thing I would do is say, practice with your group, set your standards. Some people still use the 5-point scale. And if that's what you decide to go with, or if there are certain things that you want to, put into your grade 2 category, then make sure that everyone in your lab or in your group are grading embryos the same way. And so it can be helpful to, maybe have a group of embryos, have everyone go through and grade them, just hide their answers, and then you all compare your answers at the end. And do this, every 2 or 3 months, just to make sure that someone isn't changing the way they grade embryos, and the rest of the members of the lab don't recognize it. And believe me, that happens. That happens with students as we train them. They pick up these strange habits and we have to go back and correct them if we don't continue to practice regularly. Okay, so I wanted to go through some pictures, but first, if there are any questions about the grading and how we grade them, I would be happy to answer them. Let's see.
Can I, Jennifer, say something?
Sure, absolutely.
Yeah, okay. Let me, because there are some questions in the chat box and I'm sure you will answer all of them, but there are a few things in my mind which I wanted to share with you, having experience in MOET for quite a few years and then in IVF. One thing which I have seen is that when we are doing your talk has been so eye-opening to every one of us, because the mindset with us as a new people, we think that, really we become embryologists very fast. But as you said, giving the example of the 20 years experience and the 5 years, how difference it makes to, in case of IVF embryos, normally you don't see the morulas, we have been always seeing blastocyst and above. And you feel every embryo looks good and you are like a human tendency or whatever. You don't feel like, they throwing those embryos because there is no proper grading which has been yet learned. And that's what will come with the experience. And we try to, you try to transfer them or you try to freeze them. And then ultimately your pregnancy results go down. And another disadvantage in India is that we are not having access to the cow sites like you have in other countries. Only thing is that our team can get an experience by working on buffalo oocytes, and that's how they can do a lot of practice in grading of the embryos. Then one more interesting thing, Jennifer, I would like to ask you is, normally we see the embryo on sometimes on 6th day evening itself, and we find some embryos are really ready to transfer. And then we see them on 7th morning. And again, I mean, we would like to check with you. You see them again on 7th evening also. And even sometimes some embryologists, they get tempted even to keep them and they look good. And then they see on the 8th morning also. And then it looks like a good embryo, but actually it is not, So we will like to have your views on that as to how frequently this interval which we are seeing it. So we need your comment on that. And then another thing which I would like to check with you is this, normally I have seen with the IVF embryos, there is hardly any rejections done, the grading, very serious grading needs to be done. And with your experience, I mean, it's not really a question which can be answered as an average, but what sort of the embryos, like when we do in vivo work, you see a lot of degenerates and, unfertilized, which you don't see these into the IVF embryos. In vivo, you see all sorts of these differences are there, unfertilized one and degenerated. But in case of when we look for the IVF embryos, it doesn't happen that you don't see many of these such type of embryos. So all this put together, and then really this, if you can further, in your talk, I don't know whether you'll be able to give us few minutes in sharing your experience on the biggest problem which our country faces is this, because of the logistics, taking these fresh embryos, because we have got the people who are holding 1 cow, 2 cow, and then carrying these embryos on for transfer. It's not a big recipient herds. So the freezing is the only way, even for the IVF embryos, but that's where we really we are getting very poor results. So if you have time, and then if you would like to throw some light on that. So this is in short what I wanted to ask you. And then there are some questions in the chat box. So then you can go ahead with this, please.
Sure.
Yeah.
I will see if I can capture all of that in my answer, and feel free to jump in if I miss something. So to the first part of your question, where you were saying that you see embryos that look good on day 6 or day 7, and then by day 8, maybe they don't look as good. I think our philosophy is if you're going to transfer an embryo and you have it in an IVF system, once you get to, you can transfer day 6, and if we're talking day 6, we're talking morula, you get better pregnancy rates. You tend to get better pregnancy rates with the blasts and the expanded blasts. And so as you get further on and you get towards the hatching and the hatched, even in vivo derived embryos, there's evidence that your pregnancy rates will decline. Now, we don't really know why that is. It may be just that our IVF systems are not supporting those more advanced hatched embryos as well as they should. And in all honesty, the best place for an embryo is in a uterus. So, if you have a good quality embryo and it looks good on day 6 and it looks good on day 7, I would transfer it. I wouldn't hold it to day 8 for any reason, because we do often see them decline once they hatch and try to continue to grow in vitro. And I think that's just, a failure of us to optimize our system for that kind of stage. Now, people are getting much better at growing embryos out to the 14-day point, but that's been done in mice and some other research species, but that's not really something that we're doing actively in bovine embryos, or that I would suggest for a program that's going to be transferring embryos. As far as your question about some of the degenerate embryos, I think for us, at least in our IVF system, we evaluate embryos at 2 points. So we have a 2-step IVF system where we have our maturation and our fertilization. But then for the culture portion, we have 2 media. And after the first 3 days, we go in and check the embryos for development to at least the 8-cell stage. because that's where they should be. And then we switch the media to something that has a little bit more basically energy sources as the embryo ramps up its growth. And so for us, we have cleavage rates. Now these are straight from the slaughterhouse locally here. So it's a very different source than what you would potentially be getting or what you would get through OPU, but our cleavage rates are typically fairly high. So, probably around 75, 80% of the oocytes that we put into our IVF system reach the 8-cell stage. Now, of course, you've had some really great speakers in this webinar series. And so you've probably talked about kind of what's happening. And at that point, of course, the embryonic genome takes over, and some of those embryos are going to fail to make that switch. And so they will not continue to develop past that stage. And so that's where we see more of a loss, really, at least in our system, of like from cleavage or that 8-cell stage to the blastocyst stage. And some of that, just might be attributed to that failure to convert over to that embryonic genome. So that's where we see some of the loss. And then just in general, we see more fuzzy embryos. We see poorer organization in the IVF embryos. We see it more frequently than we do in the in vivo derived embryos. I mean, with the in vivo embryos, like, when you flush embryos, in my experience, I mean, it varies very widely from cow to cow. But, in many of the cows, you get a bunch of really good embryos, and then you get the unfertilized, And so there aren't a lot kind of in that in between, because you got to think like those embryos are growing in the ideal environment where they are supposed to grow. And our IVF systems just don't have all of those things keyed in just perfectly to get everything that's fertilized, or the majority of things that are fertilized, to the blastocyst stage. So we do see some degeneration of embryos throughout the IVF process, whereas that's less common in the in vivo embryos. I hope that answered your question.
Yeah, definitely, yes. You may have a look at the chat box if you want, or whatever way you want to do. Go ahead. I mean, there are some questions in the chat box. If you would like to answer them and proceed, or you want to do it at the end, it's your choice. Yeah.
Yeah, there are a lot of questions in here. What is pregnancy percent by transferring blastocysts versus hatched? Now, I don't have the numbers. I can definitely provide you those data. I know I have some very large data sets, but I think they drop off rather significantly, maybe by 10 to 20 percentage points for hatched versus earlier stages. And so the questions about day 8 embryo transfer, at least in our experience with our cattle here, that's not a day that I would aim for. You will see lower pregnancy rates with that. Sometimes we do see blastocysts with very small ICMs, and we do downgrade for that. An embryo really does need to have a clear ICM with enough cells in that ICM that even if some of those cells die, you're still going to get a viable fetus. So a poor or small ICM should be downgraded to at least a 2. And if there are other abnormalities, I would give it a 3. The oocyte to embryo blastocyst stage in non-stimulated Holstein or Friesian heifers and cows, that I don't know the answer to. I mean, generally in IVF systems, you tend to see anywhere between, I would say, what's been reported is a 20 to 45% embryo production rate on a per oocyte basis. Now, that's really important when you look in the literature, when you look at the embryo production rates that people are reporting. Make sure you understand if that percentage is a percentage of the number of oocytes that went into the system or the number of embryos that cleaved after fertilization. So a lot of times you can read a paper and they say, oh, it's a 45% embryo production rate, but it's 45% of those that divided and fertilized. So it doesn't really give you a true representation of the efficiency of the system from a per oocyte basis, like the number of oocytes you put into the system. So that's a really important distinction to make. Can collapsed embryos result in pregnancies? Yes, absolutely. We have a time-lapse incubator in our lab, and we can see those embryos collapsing and expanding and collapsing and expanding. And we have absolutely generated pregnancies with those. So I would definitely transfer those embryos. And in our polling of the American and Canadian Embryo Transfer Associations, very, very few bovine practitioners would downgrade for collapsing. So, someone has a question about the last slide with the two embryos here, where one is a morula and one is a degenerate unfertilized oocyte. So in looking at these two, and this is a really difficult thing to describe over a webinar and when you're not sitting in front of a microscope with someone, but this you can see kind of has a bit of a scalloped appearance. You could almost resolve individual cells, but you can't really, but you can see that it has a lighter color versus this with their very dark black spots. And it's a lot smoother than this one where you can see this very clear line. And for me, when I look through a microscope, those live cells, they tend to reflect the light a little bit differently. And so this is, like I said, one of the hardest things for my students, where you have to roll the embryos and look at them over and over to try to decipher kind of which is the unfertilized, which would be this one, versus the morula, which would be that one. So yeah, having a very experienced embryologist as a resource when you're training a new group to be able to pick out these very subtle differences is really important. Let's see, different culture media and box versus benchtop incubators impact embryo quality and development. Now, this is really interesting, and I can't really speak to this from a standpoint of we've done the research and we can say this definitively. I mean, there are a lot of advantages to benchtop type incubators. And that is that they equilibrate more quickly, right? You have your plates sitting right on the heat source versus in a big box incubator, you have to wait for the air around the plate typically to get back to the temperature that you want it when you open and close a door. And those types of things, depending on how many times you're opening a door of a big box incubator, can absolutely impact the quality of your embryo. So if you have one incubator in a lab where you're running all of your clients to the same incubator and you're opening the door constantly all day, every day, you're going to see lower embryo quality. But that's because you have embryos that are going to be operating or trying to grow in suboptimal environments where the pH may be off. If if it's not humidified, you might get some evaporation. The concentrations of the components of the media might change. All of those things can impact your embryo quality from a management standpoint. And culture media, absolutely, it makes a difference. There's some good commercial ones out there. We could do a whole session just on media and media components, but it absolutely can affect your embryo quality. We have a lot of data out of Florida and Texas here in the US that heat can impact embryo quality, or at least the ability of embryos to develop. And I think what it goes back to is oocyte quality. So oftentimes if we're doing research, we will not do IVF research in the hot months of the year. So in August and September, we don't do any IVF research unless we're studying heat stress. We'll tend to push those off until October when temperatures come down and the animals recover, because we do see an impact there. Kits available for isolation of RNA from single embryos. I think there is a PicoPure kit that you can do that with. So maturation of oocytes and degeneration, that's really, it's been tricky. And, we have, I think, come a long way from the early days of IVF where we weren't very good at maturing oocytes. And so now there are a variety of different media that you can purchase, and they come with and without hormones, where you can use those to mature oocytes. But in general, you want to be able to get, a maturation rate hopefully over 80%. If you are, putting in 100 oocytes and 80% of those are cleaving, you really want your maturation, your oocyte maturation rates to be above 80, right? 85, hopefully, if your system's working well. And of course, it's very hard to see whether or not oocytes have been fertilized in a lot of our cattle species because they're really dark, right? So you can't resolve pronuclei after fertilization and those types of things. And sometimes you can see polar bodies. So if you get the first polar body extrusion, then you potentially have a mature oocyte, and so those things can be really difficult. But yeah, maturation can be a challenge, but I think there are some good commercial options out there. Extrusions in embryos are typically happen when you get cells that something went wrong in the cellular division. So they may not have the right number of chromosomes, or there might be something else that has gone wrong in the growth of that cell or division of that cell. And so a lot of times they will be kicked out of the embryo, or maybe a cell just fails to divide and it's maybe not dividing on the right schedule. And so it gets kind of kicked out of the embryo proper too. They don't seem to be harmful to the embryos, at least the live cells. So when the embryos grow and hatch, a lot of times those dead and degenerate cells just kind of float off and then degrade somewhere, but they don't really affect the embryo proper that we're aware of. And yes, it is a reason to downgrade if you have a lot of extrusions. And so I would refer you back to that IETS scale where they give you the percentages of the material within the zona that should be part of the viable embryo proper. So,
Jennifer, I have one more question. Just want to ask you, like
sure.
with the IVF embryos are there any research paper or something like that, where you say that what sort of a percentage embryos are rejected and they are not being suitable for freezing or for fresh transfer? Any idea? I mean, that sort of any comparison between the in vivo produce embryos from the same donors and then they are subjected to the IVF protocols, and then any sort of what sort of rejections normally IVF embryos are being discarded because of this? Just it could be different from donor to donor, and everything will be different depending on the oocytes and all that. But on an average, as you have been interacting with the bovine practitioners, any idea about that?
Yeah, so like the number of embryos you create that aren't suitable for embryo transfer, that's
Yes.
really hard data to get at because there aren't places where we're reporting those numbers. I think a lot of veterinarians track it themselves, but there haven't been any kind of large studies to compile all those data that I'm aware of. But what I will say is that, I mean, you've mentioned freezing quite a bit, and I know that embryos of poorer quality, for whatever reason, using the scale that we've talked about, they don't freeze, they don't survive the freezing and thawing process as well as the top quality embryos.
Right.
So a lot of times, when I have been on farm working with veterinarians and owners on these cattle ranches, sometimes what they will elect to do is transfer all of the grade 2s and freeze all of the grade 1s to maximize the number of calves they would eventually get out of the group of embryos, right? Because you will have more of the grade 1s survive the freeze thaw process than the grade 2s, essentially. So I have seen that strategy be employed on farm, and I've seen veterinarians here in the US recommend that to their clients. Now, when you get down to a grade 3, boy, it's really difficult to tell a client like this has a good chance of making a pregnancy when you freeze a grade 3 embryo. It happens, it's not zero, but it's significantly lower when you freeze thaw those really poor quality embryos. So
again,
yeah.
It's not related to your topic, but just a quick wanted to know, what in the United States, is it more freezing with the slow cooling or the vitrification followed overall?
So my understanding is that most people are still doing slow freezing because it's easy and they have the direct transfer offer. Like, that's the option. And There are some places that do vitrification. Of course, that requires the veterinarian to, have a scope to manipulate the embryos, to warm them in the correct way, which when you're vitrifying embryos, it's challenging because you have very short time periods to do these very, very specific steps.
Yes.
And to be honest, most of the errors happen in the warming process. So people not moving the embryo into the warm media as fast as they should. And they actually get ice crystals, in the cells of the embryo because they're not moving fast enough. And so technically, it's just a lot more challenging. And I think the people who do vitrification and have very skilled embryologists on hand do very well with it, but it's not a direct transfer process. So a lot of folks don't employ it currently. Hopefully that changes. So,
Thank you.
yes.
Thank you.
Let's see, you said it's not necessary to downgrade the embryos with vacuoles as some animals are producing like that, but in later slides you downgrade. Yes. So I put that up there just to start the conversation. Traditionally, we have absolutely downgraded for vacuoles. That's true. But we are now starting to hear some of this anecdotal evidence from people like Daniela Demetrio, who generates a lot of IVF embryos, primarily with OPU from dairy cows. And she is saying, we've been downgrading for this for a long time, but that may not be necessary. So you're right. I did contradict myself a little bit there, but I think the standard is to still downgrade for vacuoles. Now, if you keep good records on your animals and you know which females produce vacuoles and make decent embryos that you can use to create pregnancies, then I would definitely not throw away those embryos. I would transfer them. But that's a question that needs more study. One side seeding or two side seeding and slow freezing, it doesn't matter, to be honest. I know people who do both and they get the same results, whether they do at the top of the straw or the top and the bottom of the straw. I think the key to seeding is to make sure that you seed in the correct place. So you want to make sure that where you seed is not exactly where the embryo is, because you get the start of those ice crystals and you don't want them to hit the embryo right away, right? Like you want the embryo to be far enough away so that if you seed up here, the ice crystals can grow slowly down to the embryo and dehydrate the embryo by, taking away water from around it so that you get proper freezing. But people do both work. The frequency to collect embryos from one animal, if you're doing in vivo flushes, I'm guessing that's what is being asked. Boy, that really depends on what your protocol is. I think roughly you could do it once a month, but some people will collect a female repeatedly and then give her a long break. But yeah, I think that's, at least that's what I know. I would have to talk to a few people to see what they're currently doing. Yeah.
Thank you.
All right. So how are we for time? We're about.
I mean, we have no problem as far as time is concerned. But we are such a wonderful opportunity. We are getting all of our group to hear you. So if we have some constraint of time, I mean, you can decide it. But otherwise, we are very eager to hear you further in your presentation. If anything is left here,
Yes, I will keep going then.
Please, we will highly appreciate, Jennifer, because not everybody will get an opportunity to come and watch you, personally into the conferences. So there's a golden opportunity for each one of us, and my members will be very happy for that. Right.
All right. Great. Well, what I thought we would do is go through a few pictures. And these are pictures that I have shown at conferences, some of them, not all of them, but usually what I do is I ask first, what stage is this embryo? And then I ask people to grade it. Okay, so you are more than welcome to throw it in the chat. I'm going to open the chat box just to see if there are any answers. So first, if we think about like what stage of development is this embryo? When I look at it, I see no perivitellin space, okay? The blastocele cavity has pushed those cells all the way out to the edge of the zona. It's touching it all the way around, so it's not an early blast. And the zona is still thick. So it is a blastocyst. Okay, so in the staging system, it would be a 6, right? A 4 is a morula, a 5 is a early, a 6 is a blast. So this would be a 6. And then what day is this? What grade is this day 7 IVF embryo? I've got a 6-1 here. And 6-1, yeah, absolutely. I would agree. This is a really, really pretty IVF blastocyst. You've got a clear ICM. You have a trophectoderm with almost no abnormalities whatsoever, at least none that we can see. I would call this a 6-1 every day. Okay, so what about this one? So you can put in a 4 for a morula, a 5 for an early blast, a 6 for a blast, 7 for expanded, and then give it a grade as well. This is an in vivo derived embryos, so IVD. Okay, so if it's an in vivo embryo, how would that change it? Okay, we've got 5-1, 5-2. This is a really tricky one. So from what we can see in this picture, and this is the challenge with grading with pictures, is there could be a blastocele cavity here sitting on top because it's a little bit blurry, but we don't really see, like, maybe that could be a blastocele cavity, but it could also be like a clearer space in one of the cells. So we tend to give it a 4, okay? And that can be a grade 1 in an in vivo system. Okay, so morula are totally normal on in vivo day 7. And then a 2, because of some of the extrusions, is what most people have given this when polled live. So we would call this a 4-2. Now, if the cutoff is 15% for grade 1, these 2 blastomeres that have been kind of kicked out of the embryo here, they're right on the borderline. So I will say in some seminars, people will give this a 4-1 on day 7, but I would tend to call it a 2. Okay, this is a day 7 in vitro produced embryo. Okay, what would you, let's stage and grade it. Go ahead if you want to throw in the stage and grade in the chat. Take a look at this, see what you think. Does anyone like this embryo? So we've got some 5-2s, 5-1. So this one's a little bit tricky because of the picture. So if we look at the thickness of the zona, it's got a little bit of a halo from the microscopy in the picture we took. So if you look at the thickness of the zona on this one, now if we go back and look at the thickness of the zona on this one, it might give you a little bit of a clue. So a 5 would be an early blast, which there's perivitellin space, but because the zona has thinned out, we would call this an expanding blast typically. And because of kind of the disorganization of the ICM, and we have some really dark spots here, there is an extrusion out here, but we've got these kind of cells that are abnormally shaped and sized in the trophectoderm. This one is often graded as a 7-3 live. And like I said, the 5 that a lot of people are saying makes sense because of the perivitellin space, and the thickness of the zona here is a little harder to resolve, but it's very thin. Okay, what about this one? This one I pulled from the IETS bovine ova tutorial online, which if you're a member of IETS, you can absolutely access that. Has a lot of great pictures. If it's a day 7 in vivo derived embryo, what would we call this? Looks like a 5-1. Yep, 5-1, 5-1. Yep, okay, 5-1. I agree with all of you that I would call this a 5-1. Okay, what about this one? Also a day 7 IVD embryo. It could be IVP as well, but it is actually an IVD. Okay. Well, I would personally also call this a 5-1. Okay. We've got a 6-1 here, a 5-2. All right, we've got a little blastocele cavity here. Okay, so that makes it an early blast. So that's a 5-1. Now here is how IETS grades these now. So this was the first one I showed you. This was the second one. And remember when I went through the staging and we talked about how if the blastocele cavity is at least 50% or more of the embryo proper, then IETS classifies it as a blastocyst. Well, this is how they classify it for stage. Okay, so they call this a blastocyst and this an early blast. And so this is one of those areas where there is a lot of kind of divergent opinions about what these two embryos are. So for me, in my lab, we call this an early blast, but according to the IETS bovine ovids tutorial, it is a blastocyst. And I think it's very difficult to to create that line for yourself of like, this is 50% or more. It's clear this one isn't, I think. So early blast makes very good sense for this one. But this one is where when I have polled people live at seminars, it's a 50-50 split. Some people really follow the IETS guideline to a T and say, nope, it's 50%, it's a blast. And the other half call it an early. So this is one of those embryos or those stages where you're going to get a lot of different opinions out there. Okay. What about this one? Stage and grade. This is an IVF embryo, day 7. 7-1, 7-2, 7-2. Yeah, so we've got a lot of 7-2s coming in, 7-2s. So here what you should hopefully see is the inner cell mass here. And then we have this darker area right here. And these are extruded cells that are squished against the zona pellucida. The zona is thin, so it's definitely an expanded. It's a 7. And we have some minor imperfections potentially in the trophectoderm. It's a little bit hard to resolve. But this is one of those, and I wanted to show it to you because I would probably tend to give it a better grade for the really nice ICM and the consistency kind of the trophectoderm here and those cells not having a lot. But this right here, the question is it 15% or more, right? That would make it a 2. And it's really hard. So this is another one of those embryos that often gets given a 2. But I would say that the quality of the ICM and the evenness of the trophectoderm cells, you could argue very strongly that this would be a 1, because when this hatches, those cells up here are going to go away and you're going to be left with a really, really pretty embryo. All right, what about this one? A 7-1, 7-2, 7-3. Okay. A 7-1, 7-3, 7-3. Okay, so here's what I see when I look at this embryo. We have a blastocele cavity, okay? The zona is thinned. It's thinner than normal, so you could definitely call it a 7. Whether or not you call something expanded, even though it's not fully expanded, is also a point of not contention, but where people often differ. So you have a blastocele cavity here. The problem with this embryo is that you can't really discern an inner cell mass. All of this stuff here is outside of the embryo proper. So we definitely don't really have an inner cell mass anywhere along in here on the trophectoderm. Maybe it's under here. So I would call this a 3, a 7-3. Yeah, because you just really can't see that nice ICM. All right, this one's a video. I'm gonna start this video. And then if you wanna put in a staging grade into the chat as you look at it, I'll play it. I'll play it once and then I'll let you see it again. It's a slow moving embryo. It's like, yeah, just kind of rolling on its own in the dish. All right, so you're about to get the final view of it here. Okay, I'll roll it again for you. Got a 7-1 collapsed, a 7-2, a 7-1. A 7-2. So you guys are right on target. It is absolutely a 7, okay? And it is collapsed a little bit, so you've got that. I would say this is a 7-2. So there's especially in this last view here, you can see there's quite a bit of extruded material that's obscuring the view of the embryo proper. But in this view here, if you look in this area here, it looks actually like it has a very nice trophectoderm. A lot of these cells are even in size. And I think some of these kind of darker spots here are actually not part of the embryo proper. And then you also see this very nice ICM right here. So I would give it a 7-2. Okay, here's another video. Okay, we'll do stage and grade. And it's an IVF embryo. Okay, any thoughts? All right, so as far as the stage, what do we think this is? Okay, we've got a 4-2. Okay. We've got a 7, a 6. I'm gonna hit play again, just in case anybody wants to get a second look at it. Got a 4-3, a 5-2. This one's tricky. It is tricky. Okay, so this is a morula, so it's a 4. Here is your embryo proper. All right, so if it's an IVF embryo, it's automatically a 2. Okay, so without even considering the other morphology, it's a 4. On day 7 in an IVF system, so we downgrade it, okay? And then you have this one big piece of extruded cellular material. So there are maybe some other small pieces here, but if you look at it, I'm gonna pause it right here. You've got this big bit of extruded material here. I think it fits into the category of a grade 2 for morphology as well. So I would also give this a 4-2, like some of you out there, because if you look at this morula, it actually looks fairly healthy. It looks like some of these cells just stop dividing very early on. So I think there's a good potential for this embryo to continue to develop for transfer. All right, what about this? There's no video here. So unfortunately, you're just stuck with this view of it. Got a 6-1, a 6-2, a 7-2, a 6-2, a 7-1, 6-2. All right, so I would first of all agree with everyone who says it's a 6. All right, the zona is still quite thick, so it has not started expanding yet. So therefore, I wouldn't give it a 7. There's no perivitellin space, so it's not a 5. It's not an early blast. And then from the standpoint of what the quality is, so it looks like there's an ICM here. We've got some of the blastocele cavity here, some of it here. it's a picture, so we can't tell if that blastocele cavity connects across the top of that ICM. We would presume that it does, but you can get double blastocele cavities in embryos, which would be abnormal, but they also still result in good pregnancies. And so I would probably give this a 6.2 as well. There's some abnormalities, some small vacuoles in this embryo, but yeah, I would agree with a 6.2. I will say in other seminars, there's been a split between 6.2 and 6.3. But I would probably be a little generous with that one. Okay, I'm only gonna do 2 more after this one for looking at embryos together. So what about this one? We got a 6.3. So the first thing to note is the zona is thick. Okay, so it's definitely not a 7, right? We've got a 5.2 and a 4.2. Okay, so what I see when I look at this embryo is an extruded blastomere here. We also have some sort of extruded cellular material over here. So your embryo proper follows this line here, and it looks like there's a hint of a blastocele cavity. So I would give it to anyone who said 4.2 or 5.2, because it's really difficult to tell from this picture whether or not this is a blastocele cavity. If you could confirm that by rolling it, I would definitely give it a 5.2, and if not, then I would give it a 4.2 if it's not a blastocele cavity. Okay, I'm going to show this one really quick. Okay, what is this? There's a raised hand. If you would like to ask a question, feel free.
Yeah. Mohanty, you want to ask something?
Maybe it was an accident. They were just waving at me. So hi.
Okay, yeah.
Okay. So we've got 4.2s, 5.3s, 5.2. Here, right here, that's a little blastocele cavity. Okay, so it's a 5. And I would say based on the amount of extruded material that obscures the top of the embryo, or obscures the embryo here because it's on top, this would be a 5.3. So, okay, one more. How about this? Degenerate, we've got a degenerated there. This can be a tricky one, particularly if you aren't looking at an embryo at a higher magnification. Okay, so the person who said degenerate is absolutely right. Okay, what can be tricky is that this would be classified in most cases as an unfertilized. So sometimes oocytes can fracture, and that might be what this is. These are really small to be true cell divisions and to be early blastomeres. So for, it could be like one blastomere that then divided again, but a lot of people would just call this a UFO. But what can be really tricky is that when this cell started to die and the cytoplasm began to fall away from the plasma membrane, it created a space that some people might mistake for a blastocele cavity. So be careful of that, okay? This is just a collapsed expanded. I just wanted to show you this one and how it differs from a morula, because a lot of people would look at this and say, hey, there's PV space, but the zona is a little bit thin. And you can't resolve individual cells really. There might be some that are being extruded here, but also it's collapsed down to one side, which you more often see in collapsed expandeds rather than in morula. Morula tend to have even perivitellin space all the way around. Most of the time, okay, there are always exceptions. Okay. So unless there are any questions about those embryos, I'm just going to mention a few final things before I wrap up here. And I think it's always worth considering how we can be better at grading embryos and how we can better predict which embryos will result in a pregnancy. And it's worth acknowledging that, you know, the scale we use for IETS and the way we grade the embryos was established with in vivo derived embryos. And there may be some features of the IVF embryo that warrant us rethinking some of those things. So for example, we've talked about the vacuoles today, which there might be some evidence for that they are not a reason to downgrade. We've talked about the shape of the inner cell mass. You know, what really is what we're striving for and what really makes a difference, whether it's like round and compact or maybe a little bit more spread out. And I think it's also worth looking at other systems for grading embryos and how they do it. So I teach this master's program in assisted reproductive technology, and a lot of my students go out into the human infertility world. And so we also teach the Gardner scale for grading human embryos in our lab, and we do it with bovine embryos. But what's interesting about this particular scale is rather than assigning one number to the whole embryo, the grade of the embryo has three components, okay? So they grade the stage, or they evaluate the stage, they give the ICM a grade, and they give the trophectoderm a grade, okay? So this is just a picture of a pretty nice human embryo. Okay, so the grades vary slightly. So they have kind of this, they don't have like the early blast and the blast. They have a blastocele cavity of less than half the volume, a blastocele cavity of more than half the volume, and then the full blastocyst is where it's touching the zona pellucida all the way around. Okay, so that's one of the differences in this particular system. But then they have expanded, hatching, and fully hatched. So very similar to what we do. Now for the ICM, if it's a perfect ICM, it's compact, there are a lot of cells, it's an A. If it's loose, kind of a small ICM, like someone mentioned earlier, that would be a B. And if it's hardly resolvable, very few cells, they're very disorganized, and you would give it a C. And then for the trophectoderm, if there are a lot of cells, they cover the whole blastocele cavity and they're even in size, there's no abnormalities or irregularities, then you would give it an A. If you have some cells that are a little loose, or maybe some that are a little bigger, a little smaller, you might give it a B. And then if you just have a very poor ICM with some really big cells that stretch across that blastocele cavity, you would give it a C. Okay, so just to kind of throw some of this at you with some grades, A, really nice inner cell mass, a B. So if you look at this trophectoderm, you have cells of different sizes. So you've got these rounder cells here, you've got some longer cells over here. So there's some irregularity in the trophectoderm. Here, the ICM isn't as well organized. So here where everything looks very uniform and compact, in this particular embryo, we've got some bigger cells and smaller cells, and it just doesn't look quite as cohesive. And the trophectoderm, again, the same as this one where you have cells of different sizes, versus this hatched human embryo where you've got this beautifully compact inner cell mass, and look at all these trophectoderm cells. They all look almost the same size. They're very uniform, and you have very few problems there. So, you know, maybe this is something for us to think about when we're grading bovine IVF embryos, is should we think about assigning a grade to the inner cell mass and the trophectoderm? Or maybe what we say is that, you know, if there is an abnormality in the ICM, then you downgrade, or if there's an abnormality in the trophectoderm, you downgrade. If there's an abnormality in both, then maybe that's a 3. So there could be some ways we can improve our grading system there. So I'll put that out there for thought. And then I put this picture up here because on the other side of that argument is maybe we don't need a new system for grading IVF embryos, because as we get better at making them and our systems get better and we get closer and closer to the IVD gold standard, you can see that some of our IVF embryos are indistinguishable from our in vivo derived embryos. So I like to think about these arguments from both sides, and I think there's a case there to be made too. I think there are other things that we will ultimately have to consider when we grade embryos. And this was really interesting also from Daniela Demetrio, where she had these beautiful embryos she got out of a dry cow, and she found that in many of her lactating cows, she had poorer quality embryos. Again, this needs to be followed up with some papers and very strategic and targeted research to answer these questions. But anecdotally, it's out there that there may be a difference in embryo quality because of the status of the donor. The breed of the cow can also have an impact, and also the age of the donor can have an impact. So, and then just to throw it out there to make it even more complicated is that in the human world, there are all of these artificial intelligence or machine learning systems that are being generated to grade human embryos and take humans out of the equation altogether. And so they had, this last slide basically just says there were 16 papers presented at, these are the main human meetings in the US and in Europe in 2019, back when we were all getting together. Hopefully we're all getting together again soon. But there were 16 papers at those meetings that looked at different ways of using machine learning to evaluate embryos. And this is just an example of a paper where they had 15 very experienced embryologists go against a system, the system that was presented in this paper. And the system was more accurate in picking embryos that would implant and embryos that implanted that were also euploid, so had the correct number of chromosomes. So just some food for thought. That, you know, we have a good system in place. It's been tried and true for a very long time, but we can always potentially improve it. And maybe that's through new technologies, maybe it's through a new system for grading IVF. But I think, you know, I have talked about this embryo app for a very long time, because what we really need are data, and we need large data sets with, you know, embryo transfer and pregnancy rates connected to these different embryo morphologies. And so maybe it's time for that.
Thank you. Thank you. Sorry. Yeah.
Yeah, no, these are some resources for people who want to do more embryo grading. There's a chapter in this book. Here's just, you know, different places you can find information. And then there's my email address in case you have any questions after this.
Thank you very much, Jennifer. It was a really wonderful talk. And I think on behalf of the whole Bovine Embryo Transfer Group, probably I'll get in touch with you separately and maybe request you to come to India for about a week or 10 days and conduct a training program for our budding embryologist. That will be really a great thing. So for which I'll get in touch with you separately. Now I'll request Dr. Sanjeev Chaubal from United States. He's also across there to conclude this webinar. And again, once again, Jennifer, I'm really grateful to you. Now over to you, Sanjeev, please.
Hi, Jennifer.
Hi.
So on behalf of the entire group, I really want to thank you for an excellent presentation. I'm sure that, you know, you have given this kind of a talk many times in conferences with live audiences also. But this is probably a new experience for a lot of people on this group because, you know, in India, what has happened is we really have missed the bus for the MOET technology. You know, we started with AI 60, 70 years back, and then we did a little bit of flirting with MOET way back in 1990s, but it never really took off. And so we completely missed that bus. Luckily, we are again trying to catch this bus for IVF, and that's why this group is, it's a mixed bag. There are some old people like me and Dr. Zawar who were part of the first wave of MOET technology also. But most of the people are learning their ropes here on the IVF technology, and they are very, very new. So it's a great learning experience for them. I would just comment one thing that you touched upon actually on the freezing of embryos, which is a very, very important part. And actually, embryo evaluation makes a very critical part of you know, selecting which embryos to freeze and which embryos not to freeze. See, unfortunately, what is happening in, I'll first comment on what is happening in US. As you know, here, as you said rightly, we transfer mostly grade 2 embryos, or we try to transfer grade 2 embryos and try to save grade 1 embryos for freezing to be used on a later date. And still we get pretty decent pregnancy rates. In India, unfortunately, what happens is because of a lot of constraints with getting proper recipients and synchronization and small farm sizes and all those kinds of things, freezing of embryos is still considered as a very, very difficult task, because we are just not getting enough or good pregnancy rate with the frozen embryos. And so people tend to end up transferring mostly grade 1 embryos fresh.
Okay.
I mean, that is good on a short-term basis, because you are going to get some good results, but you cannot expand the business based on just fresh transfers, because it has its own limitations, especially the logistics. And so unless we switch to freezing embryos by selecting the best embryos for freezing, we are just not going to expand this technology in India. So I think that kind of gives the basis for how important your presentation is, because evaluation of embryos is the foundation for all this. Dr. Zawar has already mentioned, if you could come visit India for a 7 days or 10 days training session, that would be wonderful. But till that time, I would suggest that if we could create some kind of a platform where our people can share their photos of embryos and request your expert opinion on some of those, maybe that could also help a lot, a lot of people in improving their skills. So with those comments and those thoughts, I'm just going to conclude. Once again, thank you very much from all of us for giving this excellent presentation, and we hope to interact with you more in future. Thank you.
Well, thank you very
Thank you,
much
Jennifer.
for all those kind
We're
words.
almost given 2 hours of yours, and we are really, again, on behalf of the whole group and the whole country, we're really grateful to you, and we will keep in touch with you. Thank
That
you.
sounds wonderful.
Sorry, you're saying something, please.
I said, that sounds wonderful. And I do hope to visit India. That would be actually kind of a dream come true of mine. I've always been very keen to visit.
Yeah, we will love to have you. I mean, I'll get in touch with you, and I'm sure it will be possible. Yeah. Because, we had, probably a couple of the embryologists like Jen. I was with her in 1985, well back, and again in 2016. Jen Pryor, you may be knowing her. You know her? Yeah, Jen Pryor, she had been to India, and I worked with her back in 1985 and again in 2016 with the Charles Looney and Jen, and Yeda from Brazil, she had been here. Then Lottie from Denmark, she had been here. So there are a few embryologists like you and Daniela, which are due to come to India.
Yeah. Well, you've got it. I mean, those are all great people who would be our great resources.
Thank you very much. Thanks a lot. And have a good day ahead. And once again, a lot of thanks to you. Thank you. Good day. And conclude it like an Indian way. Namaste.
Namaste.
Okay. Thank you, everybody.
Thanks, Jennifer.
Thank you.
Webinar is a close. Thanks a lot. All my Indian colleagues also. Thank you. Thank you.
Thank you.